Welcome also from my side. So for those of you that do not know me, I'm Thorsten Reusch. I'm heading the Marine Ecology Research Division at Geomar. I do not have any personal experience with corals, but we are working a lot with other marine organisms such as seagrasses, but also fishes. And our niche, if you will, is the evolutionary ecology of marine organisms. And I've also been the founding member of the Coral Restoration Centre that's trying to do exactly what we are doing today, so to translate evolutionary concepts to adjacent fields, including conservation. And maybe also very few words on this location here. The Zoological Museum is actually biology-wise a famous place. So not only was the term biocenosis got here by Carl Möbius, so that was in a way that's the foundation of community ecology, but also plankton. The term plankton was coined here because plankton nets were built here. You will just see them at the entrance to this lecture hall by Victor Hansen about 140 years ago. So I hope that you can also breathe a little bit this very old spirit of some of the founding fathers in that case of ecology and marine ecology. And with this, I stop, and I'm very happy to introduce the speaker of this afternoon session, which is Iliana. Which is Iliana Barnes, and she received her PhD in 2004 from the University of Miami. Then spent a postdoc, a brief postdoc there. Then went to Hawaii for a more extended postdoc. Was then hired as an assistant professor at Penn State University on a 10-year track, and she got tenure in 2013. Congratulations. And she has been working ever since on the ecology of corals, starting with more, let's say, traditional assessment of gene flow and population structure, and then moving more and more, also, I guess, driven by the conservation crisis, to restoration questions, to questions of reproductive biology. And we are very curious to hear what you will contribute here to the coral, to the assisted evolution. Thank you.
Thank you, Thomas and Malina, for an already wonderful day, and thank you for organizing this symposium, giving me the opportunity to tell you a little bit about our more recent work on corals and a somewhat personal perspective on how corals may survive climate change. So what I'm going to do today is share some case studies that I think might have some implications about how we address this question. As a bit of background, you know, in the NOAA Coral Reef Watch termed 2014 through 2017 the third global coral bleaching event, with over 70% of coral reefs around the world experiencing heat stress that either caused bleaching or mortality. And in addition to this, in the Caribbean, even those areas that are not quite as deep red, then were hit by 2 Category 5 hurricanes. So we've really been through a lot the last couple of years. This is an illustration I stole from Charlie Veron, indicating the process of coral bleaching. Corals live in symbiosis with single-celled algae. Small increase in temperature or a decrease in temperature, or some other stressors as well, can cause a dissociation of the symbiosis and coral bleaching, leaving behind a thin layer of see-through tissue, transparent tissue over the white coral skeleton. And in the 2000s, folks started to really think about the long-term applications of increased coral mortality and build models that predicted the demise of reefs. And a fundamental assumption of these models was that corals cannot adapt. And you know, we have since accumulated quite a bit of evidence that that statement is unlikely to be true, even without any intervention from us. So with that brief introduction, I want to discuss assisted evolution, assisted migration, keeping in mind that I think we've more or less reached a consensus of a goal of preserving or conserving functional reefs perhaps over the goal of conserving particular species. We can discuss this later if you think that that's not the case, but in this talk, this is what I perceive as our goal: conserving functional reefs. I've been involved in the Caribbean Coral Restoration Consortium, which is broadcasting this the symposium today as a webinar, and particularly I am the chair of the Restoration Genetics Working Group, and it's one of 5 working groups with a very heavy emphasis between practitioners and researchers. So this consortium really is trying to translate research immediately to practitioners and in making information usable and available. If you, especially those that follow online, if you feel like you're all lost in the genetics, there's a couple of webinars on there that go into Genetics 101, so perhaps I can refer you to that for getting some background for today's symposium. The immediate research goals we've decided on for the Coral Restoration Genetics Working Group, again, are split between some sort of basic science research and application. And one very necessary application for our Caribbean practitioners is to have genotype databases and reliable phenotypes. This is technologically feasible. It takes a bit of effort, but we can do this, and we will in the near future. More complicated and perhaps taking much more research effort will be to understand the genetic architecture of coral adaptation, as well as developing models of the genetic impact of restoration activities on coral populations, such as how adaptive alleles may spread if there is outbreeding, depression, or you know, if that is not a particular risk we have to be worried about. The other, I think, really important point I want to make today is that I think we need to view our available options in the context of the ecoregions we're working in. And I think there are some fundamental differences between, say, the Caribbean, the Eastern Pacific, the Central Pacific, and the Red Sea. The Caribbean acroporids are severely threatened. There's, we have, 2 species rather than the hundreds that are available in the Pacific. And yet these 2 species occupy all of the niches, basically, that the Caribbean, that the Pacific acroporid, many acroporid species occupy. The life cycle of our main reef builders in the Caribbean is very similar. So this includes the acroporids as well as the orbicelids. They're hermaphroditic. They release eggs and sperm to the water column. They will not self to any extent. The planial larvae mature in the water column until they are capable of settling. When they've settled, a primary polyp develops, and that's when the simoiodinium are taken up from the environment. Budding then produces a mature colony. That's an asexual process right there. And the mature colonies often fragment for all of the major reef builders, and we get sometimes large colonial stands. Now, the other aspect of fertilization biology in the Caribbean reef builders is that we have very variable compatibility, or what appears to be genetically determined compatibility between certain genotypes. So when we do crosses between 2 parents, our fertilization success ranges from 0, 7%, all the way to 100%, and this is actually work that's just in press, led by Margaret Miller, who's in the audience here, both on Acropora palmata as well as on Orbicella, where we have average fertilization rates of, around 50% or so. So this is another important aspect to keep in mind. Having 2 genotypes on a reef does not mean that you're going to get successful fertilization. And in these Caribbean reef builders, sexual processes are often not all that important in terms of contributing to population maintenance. Asexual reproduction is a major mode of population maintenance. And this, again, induces, it is in the major reef builders, it's through fragmentation rather than production of asexual larvae. This then leads to large stands of clonal, or can lead to large stands of clonally produced colonies. So here, these are clonal maps. The shape indicates the host genotype. So at the Horseshoe Reef in Florida here, it's all one genotype represented by many colonies. And the fill indicates the genotype of the symbiont, and in this case, Acropa palmata associates with just one species of Symbiodinium fiti, and using microsatellite markers, we can determine the strain identity and know that there's high fidelity between the host genotype and the particular Symbiodinium strain that genotype associates with. It's very stable through time and space. Not 100%, sometimes you do see switches. And the clonal structure differs across the region, where sometimes you have only one symbiont strain but many host genotypes. In other cases where there's both symbiont and host genotype diversity. The same is true for Orbicella annularis and Fabulata, even though here the symbiosis is much more complicated. There's several different clades of Symbiodinium involved. And still though, the host here in this case, it's the color that indicates the host genotype. And in both cases, you can get large fields of clones, even though one is a more columnar growth form, the other one's more sheeting. This leads to sometimes extremely old genets. This is data we've produced using somatic mutation occurrence in our microsatellite data, and we estimate that some of these very old large clones, or like the large clones, can be old, ranging from hundreds of years to perhaps even over 1,000 years, and this is true for Palmata cervicornis as well as the hybrid. We have some hybrids who appear to be several hundred years old. So I want to stress that, you know, I'm not making any claims that there's fitness effects of these somatic mutations. I could see that perhaps, you know, accumulations of somatic mutations may influence successful fertilization, but we certainly don't have any direct evidence of that so far. So this brings me to thinking about assisted evolution and assisted migration, and behind this background of the biology of the species in the Caribbean. And first, I guess it is important to mention that, you know, assisted evolution does assume that the natural processes are too slow, so, you know, we have to intervene. And what I understand this to mean is that under assisted evolution, we assist corals to evolve temperature tolerance by either repeated rounds of artificial selection or by direct genome editing, versus migration, assisted migration, where we move corals to new habitats that are now becoming habitable to climate change, but that corals may not be able to reach by themselves easily. So they might eventually get to northern Florida, but, you know, we're just going to help them along and get them there a little earlier. Let me first spend a couple of slides on assisted migration before going into assisted evolution. Here, the premise is that under climate change, the habitats will shift, but foundation species may not be able to reach those habitats. And the procedure would be to translate corals and larvae to new empty habitats, say northern Florida, or translocate the corals from historically warmer sites to those that are expected to become warmer in the near future, say Red Sea to Australia. And the outcome would be to establish new populations and locations that enable future survival without further intervention. And you know, the most commonly raised concerns are that such habitats may either not be known or they may not exist, and that this may cause outbreeding depression, where the offspring of crosses from the translocated corals with the natives, if there are any, result in decreased fitness of those future generations. Michel Matz's group has just modeled this, and he thinks that there's no way we can move enough individuals to cause outbreeding depression. So if you're interested in this, there's a preprint on bioarchives addressing this particular question. In terms of the Caribbean, we think of specifically Florida. I mean, the most obvious thing to do would be to take corals here from the Florida Keys and move them northwards. And this is a plot of satellite-derived solar insolation along here in Florida. You see that it's just, there's less, you know, light basically available in northern Florida. But this is satellite derived, and you know what I would argue here is that we have improper knowledge of the relevant environmental variables to really understand where these novel habitats might be. I mean, for example, compare Mexico and Navassa. They're the same latitude, different longitude. The amount of solar insolation they receive is quite different. But this is satellite derived over land. You know, these are not measurements that are taken on the reef. So. You know, I think it's really important that we get a move on and understand the critical environmental variables as soon as possible to make some decisions of where suitable habitat might be. So this brings me to assisted evolution, and here the premise is that there's standing genetic diversity within populations with respect to temperature tolerance or other stress tolerance, and some individuals may survive or resist this particular stressor. So a procedure would be to breed resistant colonies to produce offspring that are resistant, or perhaps alter the genome to result in resistant colonies. And the outcome that we're hoping for is getting the temperature tolerance of a population or species to improve. So this brings me to my case studies, and I guess the first one is the question, well, is there standing genetic variation to select from, right? Are there some genotypes that are more tolerant than others? And the answer is yes. I mean, when we create controlled crosses and expose them to stress and look at allele frequency shifts, we can easily demonstrate a shift in allele frequency between control and heat temperatures after a period of selection. So this is a response to selection. It is a shift in allele frequencies. So there is variation to select from. Now, what we have, I mean, we can see many effects of increased temperature on larval development. There's different contribution of parents to batch cultures. The larvae develop faster, they swim faster. They also have a higher rate of malformation. We know that apparently that energy demands are highly increased, basing this on gene expression patterns, but we don't know is whether those heat-selected larvae then also enter the population, because we have a real lack of settlement and recruitment to our reefs. We have a hard time doing this in the laboratory. So the next question then is basically, are there generally resistant, stress-resistant genotypes? Some of these are known from plants where you have genotypes that use particular pathways that make them less susceptible to stress. This then is closely tied to the question of the genetic architecture of the resistance and whether or not that might be the same across species. It would be great if we had the same stress response or same pathways involved in all species. It would certainly make our lives a lot easier. Now, keeping in mind that the temperature selection we're talking about is in an organism that has a bipartite life cycle where the temperature effects may or may not be the same and may require reciprocal mutations in the associated synmyadinium. Now, this does work because, corals have adapted to higher temperatures in the Red Sea, so it's not like this has never been done before, The question is, over what time scale and how to get there, I suppose. So then if there or might there be such general stress-resistant genotypes, like here, I'm using, again, the example of Florida, because in Florida, we not just have heat stress. Before the heat stress of 2014 through 2017, we had the most severe coral bleaching in Florida because of a cold water stress event. So in 2010, temperatures on the reef dropped below 10 degrees Celsius in cases, and this caused widespread mortality. And so, the question that we had then was, especially in Florida, they can be trained to evolving both hot and cold stress tolerance, because they're getting, depending on the season, you get fluctuating selection pressure here on surviving hot and cold temperatures. So my former student, postdoc John Parkinson, on the Devices Lab, did a experiment where we collected common garden layers, so cornice colonies, before the summer max, after the summer max, in the winter minimum, and after the winter minimum temperatures, and repeatedly exposed those to hot and cold stressors and looked at gene expression as a response. And as you might expect, you'd see a variety of patterns, control genes as they're supposed to, doing nothing. And then in some cases, these are the 4 time points, expression goes up. In some cases, it goes up in hot and cold. Sometimes it does the opposite. So overall, when you count all of those patterns, the good news is that most of the responses were allied. That means the genes that responded to temperature stress did the same thing under hot and cold temperature selection. So that doesn't seem to be, the response doesn't seem to be constrained to intermediate responses, despite the fluctuating temperatures. But what would be really interesting to do now is to understand how this happened, how these gene expression patterns look like in other parts of the range where the cold selection is nearly as persistent as it is in Florida, because it just never even gets that cold. So then the question is, all right, let's take temperature tolerant or stress resistant genets and breed those to improve the overall temperature tolerance in the population. And maybe this involves breeding corals that not naturally would be able to interbreed because they're growing too far apart. And here, this is a case, again, through our recent bleaching events where we happened to look at this reef, and we knew that all of the colonies on this reef were genetically identical in terms of the host genotype, and they shared the same Symioidinium fiti strain. And yet, we have had these really varied responses, bleaching responses to this temperature stress over fairly small spatial scales. So, the question, that you to scratch your head, how is this possible? How can one colony look apparently fine, another colony, have some areas that are fine, others that are clearly aren't? And so what we did, and these are all clone mates that, as far as we can tell, are, have the same genotype of host, same genotype of symbiont. What explains this differential bleaching? And, it could be epigenetic differences caused by past environmental variation. Is the microbiome difference? There's certain other protections that other microbes other than Symbiodinium impart on these colonies. Is it some microenvironmental variability? So 16S tag sequencing clearly showed that we had 6 genets that were each represented by 6 colonies or 6 samples, and clearly the genotypes the genets all contain different microbiome communities based on the 16S tag data, but bleaching didn't explain any of those differences. We then looked at methylation, and this is using a reduced representation sequencing method. Clearly the 6 genotypes here, the 4 genotypes, each represented by 6 samples, also consistently differentially methylated their genomes, but in this case, actually, past bleaching did indicate a difference in the way that the genets methylated their genomes. So some of them, for example, the antioxidant defense pathways that have come up over and over again in gene expression studies also were differentially methylated, indicating that perhaps there was an induction of these defense pathways from prior bleaching. What we don't know is for how long these methylation changes persist, whether they're heritable, and whether those really are an explanation for the differential phenotypes we're seeing. But the outcome of this is that not knowing the genotype, not having the genetic results in hand, I would have selected one of these ramets as being bleaching resistant, and would have unlikely to have had or may or may not have had a heritable trait in hand that would have been passed on to the next generation. So I guess then the last case study I wanted to share with you is whether can we use another phenotype to select stress-resistant genotypes? So if temperature isn't that reliable in this case in the Caribbean and Florida, perhaps we can use a different one, such as disease resistance. And hand in hand, there goes the question whether unassisted evolution is too slow. So here, this is work that Erin Miller gets all the credit for. She works in the Moat Marine Lab, and she had Sauropornis genotypes that she exposed to white band disease, a major killer of Sauropornis. And the good news was that, under control conditions, we had a really high percentage of genotypes that was disease resistant. This is after the population was almost wiped out, indicating that there's ongoing adaptation, to this disease. Now, the disease risk increased 2,000-fold in bleached corals. So when you, again, expose the same genotypes under bleaching conditions to the disease, they have a 2,000 times higher risk of dying from that disease exposure. Now, interestingly enough, and there was a real trade-off, right? So the higher temperature continuously removed disease-resistant genotypes from the population. Now, interestingly enough, one genet gained resistance under higher temperature. This is a really exciting genotype, right? I mean, this would be you know, a great target for breeding, except we wouldn't have been able to select it because it bleached and, you know, wasn't disease resistant under controlled temperatures. So there's also evidence for differences across the range in the amount of disease resistance we see in its interaction with temperature. I think rather than despair, you know, the message I get from this is that, you know, our best and immediate action is to cross and as many genotypes as we possibly can and sort out who's great sometime later when we have better phenotypes that tell us what to do. So I guess, you know, that means that in terms of, you know, really practical for the people in this working group, you know, that ask me, you know, how do I manage my nursery? Is assisted evolution practical and likely to be beneficial in terms of selective breeding? There's some good news, you know, there's plenty of variation to select from. There's some bad news. It's really difficult to know which ones are resistant genotypes. And, you know, that some of these temperature stress, and they don't seem to be these generally stress resistant phenotypes in this species, in this context. So again, the practical solution seems to be breed as many gens as possible in as many places as possible to assist what we can show to be ongoing adaptation in these species. And this is, of course, what SECOR's goal is. So my last point is that, you know, in the Caribbean, you have, again, a situation where I think there's another route that could lead to rapid adaptation. This is hybridization. These corals, Silvercornus and Palmata, form a natural hybrid that's termed a crop prolifera in some cases. And this was thought to be sterile. So it's thought to be that this is an F1, there's F1s that form, but they can only backcross with one parent, not the other, and there's no F2s. The good news, I think, is that genomic evidence we've just gathered indicates that there's both F2s and backcrosses with the other parent. So, you know, there's an evolutionary future for this. And the hybrid has characteristics that seem to be beneficial in terms of it being able to grow in a wider niche. It can withstand or bleach less than, or bleach to the same extent as the parents under more stressful conditions. And so, and the hybrid is less afflicted by some of the other causes of population decline we've seen. So to me, this hybrid has a lot of potential because it has a huge morphological diversity that might enable this hybrid to explore a wider niche than either of the parent species can. And it combines very different genetic backgrounds really efficiently, right? I mean, you get very different genetic backgrounds to combine into viable offspring very efficiently. So I'm you know, thinking that this is, you know, there is some real potential benefits of active intervention increasing the number of hybrids we produce in the Caribbean. So I'll close down with this and emphasize what Burke has said before, that, you know, before we do anything else, or not before, but, you know, is it very, very important that, you know, the most effective way to conserve coral reefs is to slow down climate change. And while we're working on that, I guess we could, there's some things that I think are really doable. For example, fostering ongoing processes of rapid adaptation, for example, via hybridization, as well as increasing chances of successful sexual reproduction, especially in the Caribbean. There's some mysteries we got to solve, like where are larvae going to, why they're not recruiting. You know, we need phenotypes that really truly indicate coral fitness. We just don't have right now. Understands, you know, where are Symbiodinium, how do we get the right ones into our larvae, and instrument the reef in such a way that we can really understand where suitable habitat is for assisted migration. And I guess I leave you with this thought that perhaps different ecoregions require different approaches first, because I think we do have some real differences in the way that the systems work. And with this, I think Madeline is going to give us an idea of what this might look like in the Great Barrier Reef rather than in the Caribbean. Thanks.
Thanks a lot, Diana. And
I try to understand sense when you said that you don't have phenotypes to identify resistant genotypes. I mean, is it because you lose them when you identify them, they bleach and die, or I didn't quite understand what you meant by that? What do you mean because the disease-resistant genotype was not bleaching?
No, it's because we, when you look at that field of clonal corals, and some of the ramets bleach and others don't, then I cannot say I have a bleaching-resistant genet.
Okay, because you have within the genotype, you see all this variation.
Yes. So this is not, of course, in some cases. You don't. Yeah, in some cases, you have a very consistent phenotype, right? In other cases, you just don't. So we just also then not really sure how to interpret the phenotype of bleaching. Is this truly something that we want? Bleaching resistance is truly something we want to select for, and this would be so easy to test in any other system. We could just say, okay, let's look at the fitness effect, who reproduces, has successful offspring, next generation. Just don't have it.
We do see very consistent pattern. If we try to call even like experimental tags, and we have the patterns that actually can just see which we is quite spots in India.
Yep.
So it's very different.
I think so. Yes. And Steve Palumi's lab just I think published a paper showing that they can very reliably predict which ones of their phenotypes are more temperature tolerant than others. So again, I think this is just making a case for realizing that we have a huge amount of variation in the life histories of these species and the way they react, and that not every approach is going to work for every species.
I think it might be related to the fact that you have such old clones that have accumulated somatic mutations that you actually cannot no longer the same genotype.
Yeah, we also looked at the somatic mutations that we can measure at the time, which were in the microsatellites, and there was no correlation, but that doesn't mean anything because, you wouldn't expect a mutation in the microsatellite locus to be associated with any particular phenotype. So this has to await more, genome-wide analyses, and, it's a possibility, right? I mean, these are very old, they clearly have even if it's not genetically based, it could be very persistent epigenetic changes that have accumulated over 400 years that make them really, for all intents and purposes, no longer the same.
But so long as these genetic and or epigenetic factors are heritable, it doesn't really matter whether it's epigenetic or genetic value. We actually have more than a single gene, but we have actually more than those two.
Yeah. So that's the next step, right? Figuring out, is this in any way heritable? And I just don't know yet.
Thanks, Juliana. Very nice talk. I mean, to follow on from that, it does seem that the system you're working in has had a massive selection already. I mean,
yes.
Maybe that lost 95% of the Caribbean. So, and that was disease, and again, it's more or less a single event, and I don't think, for that sort of selective pressure that we're looking for, what we're looking in the future for, and we can, as human beings, we can predict that we're going to see this steady increase in bleaching events, but the corals aren't experiencing that in a particular location at a particular time. That experience is a catastrophic mortality, and then nothing happens for 100 years in most locations. So you're not seeing that sort of environmental pressure that's going to lead to that selection. But I just wondered to what extent do you think that sort of catastrophic selection that's happened in this population particularly means that you've just got very little variation left in the population?
Well, I mean, it depends on what you're, yeah. So, I mean, I have to make clear that in terms of allelic diversity, of genetic diversity, we cannot show a bottleneck effect yet. I mean, it's unlikely that you would. I mean, you wouldn't expect there to be, right? Now, this is different than losing genotypes. We know we lost a ton of genotypes. So, particular combination of alleles in particular, ways, those we've lost, but we probably lost some alleles, but not catastrophically. And these are extremely heterozygous. So, they're not in any way inbred. though it is clear that they have undergone, you know, strong selection, and we've lost, you know, a large portion of, and we see ongoing adaptation, I would argue, given that we have a rise in disease you know, resistance in the population. So, you know, I do also see that the selection pressures that these corals experience do operate on very different timescales. These catastrophic bleaching events are, you know, spread much further apart than our annual disease outbreaks, for example, right? And so there's some questions, you know, how likely is it that we're going to be able to combine temperature resistance with disease resistance in the same genotype? I mean, there's got to be some around that are both, you know. We just, you know, I just don't yet know who those are, right? And if we do find them, is the disease resistant in silvercornis the same as in palmata? I mean, you know, it'd be so nice. We could just have one resistance locus.
So thank you, Diana, for this great
hi.
talk. So my question is a simple one.
Yeah.
Why is bleaching patchy? You would think either it's too hot or not, right? And I see no patterns, I know, and I remember at Kiel back then there were people who were measuring microgradients on the reef. What can you tell us about that?
I mean, clearly, you know, if I think this is a really instructive photo in many ways. You know, I mean, I would have thought that this means they're living in the same environment. And, you know, yes, there's some shading in some parts, and some others aren't as shaded. You know, there's some there may be a vertical relief of a meter or so, but I mean, to me, what this says is that whatever it is that varies in the environment causing this, we are not measuring, we're not seeing, we don't know what it is. I mean, is it flow? You know, is it micro environmental differences? Is it something else? Is it past history? I mean, and that's why I said, and this is probably the best controlled situation that I know of, where we can exclude the host genotype, barring somatic mutations, as a cause. As well as Cymidinium diversity, because there is no Cymidinium diversity. And the microbiome doesn't vary consistently. Now, you know, this is 16S tag sequencing. Perhaps if we had done metatranscriptome or metagenome sequencing, we would have found some rare member that imparts some difference. But, you know, I think that this just says that there is some part of the system that we're not understanding, you know.
Actually, for once, I was
Yeah, I mean, you know, I just don't know, but you know, I think, you know, when you want, and genomic tools, right, GWAS would be one way of starting to get at this, but for GWAS, you need the abiotic variables that you can associate with. And to me, we don't have them. I mean, I think the Great Barrier Reef is in better shape than we are in terms of environmental monitoring. You know, we just don't have that kind of data on those scales. We just don't have it. It is not complicated. I mean, it wouldn't take much, you know, to put a few sensors on the reef, right?
This was actually a question that's on the line. Are there any ongoing efforts for
Yeah, there's been, I mean, we've tried, you know, but, you know, our sampling, you know, the association, like the environmental data we have is on a grid of like a 1/4 degree latitude. You know, what we need is a few meters. There's a large effort by the Coral Restoration Foundation, the keys to really amass a phenotype database for their genotypes. And yeah, I mean, I don't think this is particularly complicated to do, you know, it does take, we have the samples, I suppose, we have the genomic tools, we have genomes for both species now that are also, you know, annotated, and we have genetic linkage maps, and, you know, so we have the genomic tools, but what we're really missing is the environmental data on the right scale to do the GWAS with.